Okay, let's finish up today on the innate immune system. Okay, so we just touched very briefly on the innate immune system, and I'll be covering some, briefly some diseases of the innate immune system. A lot of these have to do with the inability to of bacteria to kill the bugs that they phagocytize. Okay, we discussed this already about the innate immune system. Fast, first 4 days, pre-exists, does a great job of getting rid of infections. And even if it doesn't get rid of the infection, it keeps the bacterial or viral load down so that the adaptive immune response can come up. There's no memory associated with it, has macrophages, neutrophils, dendritic cells, cytotoxic NK cells. We'll discuss them near the end. There are cytokines, interferons, complement and serum, and we'll be discussing the receptors, the broadly specific ones. Okay, they set up the inflammatory response, and they set up the adaptive immune response. Okay, so what we'll be discussing now is we'll be discussing the innate immune response, the immediate one. These guys are already here. So you have macrophages, you have dendritic cells, you have antimicrobial peptides, which I'll discuss in a minute, and also complement. You'll have a lecture tomorrow on complement. I'm just going to give you a very, very brief overview. Carl Hamby, who's sitting in the back, will do it. Yes. Hang on a second. Okay, Juan is not here. Can you hear? Is this any better? Okay. Okay. I'll try to yell. It's funny. So for the first time, I was COVID positive at the beginning of last week and everything. So I went close to 3 years without having the problem. That's why I got the glove on, not because I want to be Michael Jackson or anything, wearing the mask. I went into health services today and I told them that, there was a mistake in the test. It wasn't a COVID test, it was a pregnancy test that I was positive on. but they know me. They told me I'm too old to get pregnant, but it's okay. No, I mean, they know me. They know I'm crazy. Oh, by the way, Dr. Montecalvo, who's the head of it, she's going to be giving 2 sessions here. So she's really great. Okay, you guys able to hear? Good. I'm doing okay. Carl was nice. Carl Hamby in the back was nice enough to bring me water because he heard my voice was a little crappy. Okay. So the antimicrobial peptides and complement, which we'll discuss. Carl Hamby will talk about the complement. And then there's the induced innate immune response. By induced, it means it doesn't come up at day 0, but comes up at the end of day 2 or something like that. Okay, so it's a little bit later, but it still comes up way ahead of the adaptive immune response, which doesn't get started until day 4 and 5. Okay, so just in general, what's protecting us from infections? Okay, we have certain mechanical protections. Epithelial cells are joined by tight junctions, so bacteria don't get in. Okay. This is important because with eczema, there's increased IL-4, okay, being produced that actually will reduce epithelial cell tight junctions. That allows for staph and other pathogens to get in. So it not just is IL-4 pushing things in a bad direction, in terms of immunological, but even structurally, IL-4 is doing a very, very bad job. There's a longitudinal flow of air or fluid and the movement by cilia. This is important, okay? When Dr. Bakshi mentioned that the spike protein has to bind to ACE2, right? But if the spike protein is going by 100 miles an hour, it's not going to be able to bind, okay? So the flow, whether it's by fluid or by air, okay, the longitudinal flow will actually keep pathogens from binding to receptors on cells, okay? So that's very, very important. That's why when a person is not breathing well, when they slow down, that's not good. You lay around in bed, okay, and when you're sick, that's not good. You're probably better off getting up and exercising, keeping the flow going, keeping the breathing going, okay? Something very interesting, there's the movement of mucus by cilia, okay? Our mouths are heavily contaminated. At least my teachers told me in high school. Okay, that it was there's a lot of bugs in your throat. Okay, but your lungs are clean. What keeps them? What's the gate? What's the door that's preventing it? It turns out that our epithelial cells have cilia on them. So yes, you have mucus, and the mucus is filled with streptococcus and other stuff, but it cannot get down into your lungs because the cilia are pushing it up, and they push it up into your throat, so it actually goes down into your stomach, and the low pH kills it. This is a nice little system. Things don't get into your lung, they go into your GI tract and then destroyed there. What happens when a person has influenza? A person with influenza, the epithelial cells, okay, are destroyed. So now you lost the cilia. Forget about the cells. Now you lost the cilia that they have. So now you don't have anything pushing the mucus, which is filled with bacteria, to prevent it from getting into the lung. So what happens is the secondary bacterial infection is because the influenza killed the cilia, and now you're going to get the bacteria going down into the lung and causing inflammation. Okay, so the cilia are very, very important. Okay, there are a lot of chemical and enzymatic protections that we have. We have something called lysozyme. Okay, we make an enzyme, and what is its substrate? It's the bacterial cell wall. Okay, so can you think about it? Yeah, we make enzymes so we can metabolize food, but we also make an enzyme in our tears. You think about it, your eyes are open all the time. It should be infected more than anything else. We have lysozyme, which will kill all those bacteria that try and get inside there. Now you have the antimicrobial peptides and complement that we'll speak a little bit more about them. Antimicrobial peptides, we have peptides which will actually kill bacteria when they come into us, okay? And the great example, and different parts of our body have different concentrations of it. You know, when you pick when you're nervous and you're picking your fingernails and everything, your cuticle, you tear it, next thing you know, for the next 2 or 3 days, it hurts like hell. Okay, when I was a kid, I never stopped talking, just like now. I don't ever grow up. I saw I just read that youthfulness is not measured by genetics, activity, or diet. It's by immaturity. Okay, that was a Hallmark card that I saw. So that's what I think. So, and I was always talking when I was eating. So I get this pizza, I bite into it. Meanwhile, my tongue would be in the way, and you get this wicked, wicked gash that's just bleeding all over the place. The next day, it's gone. No inflammation, nothing. And meanwhile, I just said the mouth is contaminated with bacteria. So why? You ever wonder why if you bite your tongue, it heals without inflammation? You got antibacterial peptides, which are preventing it. Okay? So this, I mean, there's science behind all this stuff that you just haven't been paying attention to because you've been too busy focusing on the pizza. Okay? And then the complement. Okay? And what's it called? And then you have the normal microbiota. Remember I said that the pathogen has to attach to a cell, to receptors on cells to initiate infection. But if you're going to be loaded with good bacteria, okay, your normal flora, then the pathogen won't be able to, to get to the epithelial cells. But then when you have antibiotics, that's how you get the C. diff and the Candida infections, because you lose all the good bugs, and now the bad bugs are able to go in. Here's the antimicrobial peptide. Don't sweat a lot of the information. The book has tables on it. You don't need to memorize individual names, but this is what it is. Antimicrobial peptides, they're amphipathic, okay? They're able to insert into the membranes and they form pores, okay? And when you have a pore like this, you have the solutes going out into the environment and you can get water coming in and boom, the bacterium explodes, okay? This is what happens in your tongue, okay? And in other body parts also. Okay, some of them are constitutively expressed, some are inducible, and different defensins are against different pathogens, and they're made by different cells. IL-4, again, with eczema, okay, your antimicrobial peptides can be downregulated by IL-4. That cytokine that's found. So when you have a dysregulated immune system, you're making too much IL-4, okay, I said before, what happens is that you lose the cell-cell adhesion, okay? Filaggrin and other molecules are actually turned down. And then you also, you lose the ability to make a lot of the antibacterials. So IL-4 is a very, very bad player. And that's why, we have therapeutics. We have monoclonal antibodies against IL-4. And that's one of the therapeutic approaches that's being used. Okay, so this is really good. Okay, and with a lot of the immune system, there's polymorphism. Different people have different immune responses and different abilities to respond. Okay, complement. Carl Hamby is going to give you the big overview. The big story tomorrow. I'm going to give you a very quick overview. Complement is a set of proteins. It's a cascade of proteins found in the blood and the lymph. They destroy pathogens or mark them for destruction by phagocytosis. Okay, complement is a very, very powerful force, and it needs to be regulated. So you don't want it active all the time. When you want it activated, there's something called complement activation or complement fixation. It's called fixation because it turns out like you'll have the C3, okay, becoming activated. It gets cleaved, and the C3b portion of it will actually bind to the pathogen. It becomes fixed to the pathogen, and now the immune system knows that, hey, we got something here. Okay, so what's happening is that there are different ways to activate complement. Remember I said that the innate immune system has to be working from day 0 or from minute 0. Turns out that the alternative pathway is the first to be activated. The environment of some of these pathogens is somehow able to activate the C3 molecule, and it becomes activated. And just the presence of the bacterium can get complement started. Okay, and there are other pathways that we'll be discussing. Carl Hamby will be discussing mostly the antibody-derived one, the antibody-driven one, but we'll also later discuss mannose binding lectin and the C-reactive protein. These are other pathways to activate the complement, and they have to do with the induced innate immune response. Okay, so what did I say? I said that they either destroy pathogens or mark them for destruction by phagocytosis. So with complement C3, it binds to a bacterium. And then what happens is it binds to the bacterium and it recruits other pathogens, other molecules, and you get C3, 5, 7, everything. And then you end up with C9, and it binds to the membrane of the bacterium and forms pores. It's called the membrane attack complex. So just as I said with the antibacterial peptides, this is able to form a pore and it's able to kill the bacterium right off the bat. Okay, so that's one thing that complement can do, and this is something that it can do seconds after a pathogen enters our body. Okay, then there's also something, it's also able to opsonize pathogens that increase phagocytosis and recruit inflammatory cells. This is cool, okay? So here's the C3. When it's activated, here it's bound to the pathogen, but how does the neutrophil know where the pathogen is? It turns, this is C3b, the C3a is actually a chemoattractant, and it pulls the neutrophil over, and there's a complement receptor on the neutrophil, and it's able to phagocytize. Okay, and it works. This will work better with antibodies. Phagocytosis works better with antibodies and complement, but without antibodies, this is still pretty good by itself. So that's the complement system. Okay, that we're going to have a lot more tomorrow. Let's look at the cells that are involved, okay? You have macrophages. They're found in the tissues. The mature forms of monocytes. Monocytes are in the bloodstream. As soon as they go into the tissue, you got yourself a, what's it called? You got yourself a macrophage. They're long-lived. I mean, there are some that will be with you for your whole life, and they will replicate slowly, but they're long-lived. Here's an interesting thing. They will produce the cytokines, and they initiate inflammation. So they're not just here to pick up garbage, okay, and to recycle garbage that's made by cells or just to phagocytize pathogens. They will actually produce cytokines, and they initiate inflammation, and they essentially guide the whole immune system, okay? It's the neutrophils are the most abundant white blood cells. They're not found in healthy tissues. They have to be recruited from the bone marrow. They're attracted by certain cytokines, right? The cytokines are made by the macrophage. We'll discuss which cytokines attract the neutrophils. And they're very short-lived. They come in, they phagocytize the cell, they die. And actually, when you have pus in an infection, it's all neutrophils. Okay, some macrophages, but it's mostly neutrophils. The receptors of the innate immune response, I mentioned you have something like here, the CD14, which we'll discuss later, the LP. LPS is a molecule that's found on gram-negative bacteria, E. coli, Shigella, Proteus, Klebsiella, these types of bugs, Salmonella, Shigella. So all these bugs have LPS, lipopolysaccharide. This receptor and this neutrophil is now able to bind to all of them. But remember I said that, one antibody can bind to one pathogen usually, But these receptors are able to bind to many things. Maybe it's different receptors on the same molecule. It binds complement, it binds LPS, and many other microbial structures, okay? This mannose and fucose receptor binds mannose and fucose in particular orientations. We discussed the N-formal methionine receptor and the CC3, okay? You're going to see this in a lot of slides. Binding facilitates phagocytosis without inflammation, and then there's also binding by toll-like receptors that involve signal transduction with inflammation or interferon production, okay? Next. These are different receptors, okay? You don't sweat any of them, okay, except these two that are underlined. And whatever's in red is going to be important. These are different macrophage receptors, and these are the targets on pathogens. There are a lot of targets on normal cells, like, for example, red blood cells are removed from the bloodstream, okay? They're phagocytized, they've gotten rid of, right? So that's not a pathogen, and so that won't even be listed here. These are the pathogens, and these are the ligands that are seen, okay? But all these guys, including CD14, the lipopolysaccharide receptor, they just induce phagocytosis. There's no inflammation. The toll-like receptors, okay, are involved in signaling in inflammation. So there's actually different types of receptors. One is involved just in phagocytosis, and one of them is involved in, hey, you know, Houston, we have a problem. We need some inflammation to get some things done. And that's what I'm going to show you soon. So binding of receptors initiates a number of processes. We'll cover these, and Dr. Tawari will speak about this other one later on in the week. Phagocytosis, very, very simple. You got a bacterium, binds to a receptor, it's phagocytosed, okay? Then it's engulfed, right? It's engulfed, and you get a phagosome. Here's a lysosome, which has a lot of nasty things inside of it, not always, because things tend to leak, and you don't want nasty things leaking into normal cells where nothing's going on. But under good circumstances, the lysosome and the phagosome will fuse, and you're going to get total degradation of what's inside. Okay, how does this degradation occur? Degradation occurs by the production of antimicrobial peptides. Okay, and binding of the receptor induces the macrophage to make some more of these molecules which are involved in destroying bacteria. So, for example, the pH of the lysosome is low, okay, about 3.5 to 4. You begin to make toxic oxygen products and also toxic nitrogen products. Phagocytosis induces a respiratory burst, and under a fierce respiratory burst, you're going to generate a lot of reactive oxygen and reactive nitrogen species. So this is what you make: superoxide, hydrogen peroxide, singlet oxygen, hepatite, all this stuff. Okay, and that's going to be in the lysosome and then in the phagolysosome. Just something very, very interesting is here. You have NAD, these are the enzymes, like NADPH oxidases are involved, and they generate toxic oxygen derivatives. So here, look at this. Now you're getting superoxide. That's great. And not only that, but then you go from the superoxide, you have superoxide dismutase that gives you hydrogen peroxide. That's great. But bacteria make catalase, and they take this hydrogen peroxide and they make water and oxygen out of it. Okay? So we're trying to screw them, and they're trying to screw us. That's the bottom line, okay? Because they wouldn't be around unless they were able to do this. And you're going to do that assay. You're going to do a catalase assay in the laboratory. So you know that you're dealing with a worse sort of infection when the bugs are making catalase. Now, what happens if you don't make a good respiratory burst? And these are some of these, the immunodeficiency diseases. You have deficiencies in these enzymes. You have defective respiratory burst. You get chronic bacterial and fungal infections. And these are not exotic bacteria. This is E. coli, Bacteroides, everything that's in our body, even in the respiratory tract, everything. Okay, because normally the immune system keeps all these bugs down, but they're not keeping it down now. Because yes, they can phagocytize them. Okay, but they're not doing anything with them because they're not destroying them. Okay, and then there's also something called Shiozaki-Higashi syndrome, where the endosome and the lysosome do not fuse. okay? And that's the same thing that happens, okay? Just the question before, what happens with tuberculosis, a lot of bacteria, again, they're smart, they're actually able to prevent the fusion of the lysosome and the endosome. Again, anything that we do, they're going to try to stop. And it turns out that, tuberculosis, Mycobacterium, does this, okay? But when you get a T cell to activate the macrophage, it's able to overcome that inhibition. So it's a constant battle between us and them, okay? Okay, this is a table that was in the previous edition, and it had a whole bunch of immunodeficiencies in the genes. It was a great, it was a great thing to study from. The current edition has groups, not individual diseases, which I think is wrong, because this is, you guys are studying, you want to know diseases. So, for example, here it has chronic granulomatous disease, MBL, mannose binding lectin deficiencies, and it tells you what these are. So if there's a chapter in your book on immunodeficiencies, a lot of what we're discussing can be found there. Next. So we already discussed phagocytosis, production of antimicrobial peptides. Let's get to cytokine release and inflammation. The toll-like receptors, okay? So what's happening is not just you're getting a phagocytosis, okay, and destruction, but you're getting sexual signal transduction. And we're going to cover this now, okay? Remember I told you that the immune system and this signal transduction and this inflammation has to differentiate between what's foreign and what's really dangerous, okay? So what is really dangerous, okay? Something that's dangerous is something that we don't normally see and our body knows to associate it with pathogens, okay? These are all pathogen-associated molecular patterns of pathogen-specific molecules. Peptidoglycan, teichoic acid, lipopolysaccharide, flagellin, double-stranded RNA. We don't have much in the way of double-stranded RNA in our bodies. Viruses do. It's a major part. You have an RNA virus, you can be making long chains of double-stranded RNA. That's how they replicate. We recognize that as being a danger signal. When we see double-stranded RNA, okay, we know, hey, call in the reserves. CpG dinucleotides. Most of our CpG dinucleotides, okay, are methylated. Unmethylated ones are very rare. Bacteria do not have methylated ones. So when we see DNA that has the stretches of unmethylated CpGs, that's also a danger signal. This is interesting. We've already learned what is dangerous, okay? So there are a number of molecules, okay, and there are hundreds of different molecules which are actually foreign. But this is enough because teichoic acid is found on all gram-positive bacteria. Okay, peptidoglycan, cell wall, it's in 99.9% of peptidoglycan is in 99.9% of bacteria. It's not going to be found in mycoplasma or chlamydia, but in the rest of them, you're going to find it. So here's what I'm going to give you now an overview of the toll-like receptor family. This is for an overview. I'll tell you later what you need to know. Just take it in, get the, I don't like using the word gestalt. I don't even know what the hell it means, but people use it. So just get the, just get the overview.
Cells of the immune system, such as macrophages and dendritic cells, are the first line of defense in recognizing pathogens of various kinds. These cells have developed several kinds of receptors for recognizing different kinds of pathogen-associated molecular patterns, known as PAMPs. There are different classes of these proteins that recognize different types of PAMPs. Toll-like receptors, or TLRs, are composed of multiple leucine-rich repeats that are useful for recognizing various PAMPs. TLRs are membrane-associated proteins. Some are located on the surface of the cell, while others are located on endocytic vesicles, where they survey the degraded contents of pathogens taken up by endocytosis. Each member of the TLR family recognizes different kinds of PAMPs. For example, TLR5 recognizes flagellin, which is a highly conserved constituent of the bacterial flagellum. Bacterial genomes contain methylated CpG oligonucleotide motifs, which are recognized by TLR9 once the genome has been degraded in the lysosome. TLR6 and TLR2 are a dimer that recognize diacyl lipopeptides. TLR1 and TLR2 are a dimer that recognize triacyl lipopeptides, and TLR4 recognizes lipopolysaccharide, LPS, a component of gram-negative bacteria. Like TLR9, TLR3 and TLR7 are located on endocytic vesicles and recognize double-stranded RNA and single-stranded RNA, respectively. When any TLR is activated, it sends a signal to the nucleus by activating transcription factors. Not all pathogens, however, live in the extracellular space or are phagocytosed. Some pathogens, such as viruses, exist and replicate in the cytosol. There are at least two classes of receptors that can detect pathogens in the cytosol and signal their presence to the immune system. One class of such receptors are members of the nucleotide oligomerization domain family, or NOD proteins. For example, the NOD2 protein, which is located in the cytosol, can detect bacterial proteoglycans of intracellular bacteria. When the NOD2 protein recognizes its ligand, the muramyl dipeptide, it sends a signal to the nucleus to activate transcription. Finally, there is a class of intracellular receptor proteins that contain an RNA helicase domain and two caspase recruitment domains or CARD domains. One member of this family, RIG-I, recognizes double-stranded RNAs that are a component in the life cycle of many RNA viruses. This class of proteins also sends a signal to the nucleus, but unlike TLRs and NODs, it activates the production of type 1 interferons. In all, the toll-like receptors, NOD proteins, and the RNA helicase CARD domain family provide the innate immune system with the ability to detect both extracellular and intracellular pathogens and to activate an immune response against them.
Good. Okay, so some of the main points with it, there are toll-like receptors on the cell membrane on the outside, some are in the endosomes, and some are actually in the cytoplasm. There was a mistake in there. The DNA that's recognized is unmethylated DNA, like I had said before, okay? She actually said methylated DNA, so just keep that in mind, okay? Now, a lot of information here. I'm going to walk you through it and tell you what you need to know. So, for example, toll-like receptors, signaling receptors, they sense presence of infection and lead to production of inflammatory cytokines or interferons. That's what was said, okay? Now, know the association between a certain toll-like receptor and its class of ligand. And let me tell you what I mean by that, okay? Something that's found on a bacterial cell, and bacteria are usually extracellular, okay? They're going to be found on the membrane, the plasma membrane. So, for example, bacteria, parasites, lipopolysaccharide, or flagellin, those are all found on the plasma membrane. That makes sense, okay? Meanwhile, when you have things like double-stranded DNA, double-stranded RNA, single-stranded RNA, or DNA, those are going to be found in the endosomes, right? So just know that there are some in the endosomes for viruses or for DNA, and those on the cell surface are going to be for certain extracellular surfaces, okay? Now, I do want you to know toll-like receptor 4, because your attendings will probably know just toll-like receptor 4, and this way, at least, you know, you can. If you know the rest of these things, you could make the attendings on your clerkships. You know, get brownie points. But these are the. I want to tell you something about toll-like receptor 4. Okay, toll-like receptor 4 is a receptor for LPS, but it does not bind LPS. Here's LPS, lipopolysaccharide. Okay, it's on gram-negative bacteria. It binds to the LPS binding protein, and that binds to CD14. CD14 you saw on one of the phagocytes before, and showed that it bound to CD14, was able to bind to LPS. It turns out here's LPS, and then there's this MD2, which is an intermediary, and then that binds to toll-like receptor 4. So indeed, toll-like receptor 4 is the molecule, is the toll-like receptor that will respond to LPS, but it actually doesn't bind it, okay? So just, you know, that's just something to keep in mind. Okay, how does this inflammation occur? Okay, I just want you to know the major points, okay? Here you have toll-like receptor 4, okay? binding to, this complex of LPS. And here you have cytokines being made, okay? One of the major transcription factors for cytokines is NF-kappa B. You're going to hear it a lot here. Know it. NF-kappa B. NF-kappa B is a transcription factor for cytokines. And here you got NF-kappa B in the nucleus making a whole bunch of cytokines, RNAs, and then you get cytokines out. When you don't want inflammation, how do you stop this transcription? You keep the transcription factor in the cytoplasm, not in the nucleus. So here's NF-kappa B, and it's associated with the inhibitor of NF-kappa B, okay? The inhibitor of NF-kappa B keeps NF-kappa B in the cytoplasm, and you only get NF-kappa B into the nucleus when you want it to. When you have toll-like receptors being activated, binding their ligands, a whole bunch of signal transduction events happens, and you get the inhibitor of NF-kappa B kinase. Inhibitor of NF-kappa B kinase comes in, phosphorylates the inhibitor of NF-kappa B, destroys the inhibitor of NF-kappa B, and NF-kappa B goes into the cytoplasm. Why is this important for you? Well, steroids, right? When there's too much inflammation, you use steroids. Steroids regulate the inhibitor of NF-kappa B, okay? So steroids upregulate the inhibitor of NF-kappa B. NF-kappa B, the inhibitor of NF-kappa B will then keep NF-kappa B in the cytoplasm. Okay, and then there's another genetic immunodeficiency. It's called NEMO deficiency, where you have a problem with the inhibitor of NF-kappa B kinase, where you're not able to activate NF-kappa B, and you get chronic bacterial infections. Okay, so you cannot activate the inhibitor of the inhibitor so that you actually inhibit NF-kappa B. I know it makes you crazy. I can't even say it. Try and say it 5 times at the end of the day, and it's kind of rough. But that's, these are the genetic diseases that we deal with. Don't sweat the names of any of these guys. Just know that they're cytoplasmic receptors. Okay, and here exactly the same thing happens. And if here's NF-kappa B with the inhibitor of NF-kappa B, this thing gets phosphorylated, and boom, you're going to get it. Okay, and NF-kappa B sets up inflammation. Okay, so what is inflammation? Okay, here you're going to get bacteria come in, cytokines are being produced, blood vessels become dilated. Look at the difference in the diameter. The blood vessels become more permeable. You start having fluid coming in, fluid and cells coming in. You have complement and acute phase proteins, which we'll discuss in a little while. They enter the infected area, neutrophils and monocytes. Here's monocytes going through the endothelium, and then these monocytes become then macrophages, okay? This endothelium is called activated, okay? It's dilated, but it's also, it's activated. There are changes in it which allow a greater permeability, but it's selective permeability. Okay. Let's look at the actual cytokines themselves, and we'll be covering a lot more of these. This is also important in inflammation and repair, which we'll be having, you'll be having tomorrow, okay? Here's a macrophage. It's been stimulated through toll-like receptors, let's say, and you start making different cytokines. In this edition, they left out IL-1. IL-1 is a major player, okay? I photoshopped this in, okay? But it's a major player of cytokines, okay? The players in the COVID storm, anything with a star, you're getting a lot of in COVID, and it becomes dysregulated when you start making too much of this. So now look at this, okay? You're making TNF-alpha and IL-1 beta induces blood vessels to be more permeable. That's activation of the vascular endothelium. IL-6 raises temperature, and actually IL-6 drives, in COVID, drives a monocyte production, but incorrect monocytes. CXCL8 recruits neutrophils. The old terminology was IL-8, but it's CXCL8, recruits neutrophils and directs them to the infected tissue. You also get increased recruitment of monocytes, and you get activation of natural killer cells to secrete cytokines. And we'll discuss also systemic effects later. Okay, IL-1 happens to be a major problem. And just, this is from the previous edition. because this is very simple and gets the point across. I want to show you how dysregulation of the immune response can really mess up our, mess up health. Look at this. So an IL-1 is a molecule that's made. It's made as pro-IL-1 beta, okay? And it's not doing anything, okay? It needs to be activated. Now look at this, okay? You have NLRP3, you have pro-caspase. Caspase is what actually activates the pro-IL-1 beta into IL-1 beta. When a cell senses that there's damage around it or it's being damaged itself, okay, it recognizes these damage-associated molecular, molecular proteins and stuff. These are a lot like the pathogen-associated molecular patterns. This is a damage-associated molecular pattern, such as ATP. There should not be free ATP floating around, okay, in our lymph, in our blood. Well, what happens is when there's an infection and there are cells being killed, look at what happens, okay? There's ATP and it comes in. It takes these components and puts together something called an inflammasome. And then this will then activate caspase 1. Caspase 1 activates IL-1 beta. IL-1 beta goes out. It binds to the IL-1 receptor and it starts making more. So meanwhile, it's a positive feedback loop. This has to be regulated, or you could see how very, very easily you can get dysregulation. And in fact, there are conferences all over now about this inflammasome, and it's a real target for therapeutics to prevent excess inflammation. Okay, here are neutrophils in your bone marrow. CXCL8 is secreted by macrophages where you have inflammation. It makes its way through the bloodstream, gets out, through the bloodstream into the bone marrow. Bone marrow releases it. Here you have neutrophils coming out. Here's the activated endothelium. They make their way out. Neutrophils come in. They start phagocytizing bacteria. They die. Macrophages then phagocytize the neutrophils, and then everything should be fine. So this is what CXCL8 is doing. Okay, this is the last animation of the day to let you know how this activated endothelium works. One second. There we go.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. Two blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
Good.
Leukocytes.
Good. Okay. So we just saw that you have this leukocyte rolling. You have your cells actually have p-selectin in them, and then when these cytokines are released, the p-selectin is actually released, and it's able to slow down the rolling action of the leukocytes. And then when you get a combination of the p-selectin and Sialyl-Lewis X interaction, you get LFA1 and ICAM, and then you get the CXCL8. This becomes pretty much irreversible, and then it makes its way down. There are other animations on the web page. Just one point in terms of immunodeficiencies. Here's LFA1 interacting with ICAM. Another one of these immunodeficiency diseases is you're missing one of the subunits of LFA1. You get defective migration of monocytes and neutrophils, into the tissues, widespread infections with encapsulated bacteria. Okay, so all these molecules play important points. Let's take a look at the good. Let's take a look at the bad. Here's the good. Person gets inflammation in their finger. Okay, hurts, right up, increased plasma proteins into the tissue, starts getting swollen, it's red, hurts a little bit. And then one thing that you don't want, you want it to stay in the finger. You don't want it getting into the blood. Okay, blood infections are a major problem. You actually get platelet adhesion to the blood vessels, okay? So what happens is you actually get local blood vessel occlusion. You actually get clotting in the capillaries, okay? So you had these leukocytes coming out. You had the complement molecules and the C-reactive proteins and other of the induced immune response molecules coming out, and then you're closing this off. So you're walking around, you got a swollen finger, it hurts like heck, but the fluid has nowhere to go because you already blocked off the blood vessel. So where does it go? It goes through the lymphatics, okay, from the lymphatics, it goes right into the lymph node. And what's waiting in the lymph node? The T and the B cells to mount a good immune response, okay? And so that's stimulation and of the adaptive immune response, and there is survival. This is the way things should work, okay? What happens when there's a blood infection, okay? Exactly the same thing happens. You get macrophages will then phagocytize the bacterium, but where are the macrophages, okay? They could be in the spleen, in the liver. Where do they dump the cytokines? Into the bloodstream, okay? The cytokines go all over the body. So what's happening? You have systemic edema. What happens is the volume of the blood is actually being decreased because you're getting systemic edema, and it's essentially going from the blood into the tissue all over the body. What do? You put a line into the person to keep up the blood pressure, so that the brain is perfused, the kidneys are diffused, and that can go right into the bloodstream, and from the bloodstream into the tissue also. So this is a major problem. Then also I mentioned something about you can be getting coagulation, right, of the capillaries. Here you get disseminated intravascular coagulation. You get wasting and multiple organ failure and septic shock. This is what happens. Okay, septic shock kills about 250,000 people in America a year. There's no treatment for it. Okay, you just have to pray that the person's body comes up straight. But it's still it's been a problem, forever. Okay, a lot of the anti-cytokine drugs we have just don't work for septic shock, and the same thing happens with cytokines still. The distant effects of the cytokines. Here, let's take a look for a second at IL-6. Well, first of all, we know that you can get fever. Is one of them, but IL-6, these are the acute phase proteins. And this is the only thing that I'm going to be discussing here. You have acute phase protein, C-reactive protein, and mannose-binding lectin. They activate complement, and they promote opsonization. There's just one slide on this. Here's an infection. IL-6 is making its way into the liver. Liver now produces these acute phase proteins. Okay, C-reactive protein and mannose binding lectin are able to bind to bacteria, and then they are opsonizing. So the thing is, these sort of look like they function like little antibodies. Okay, they're not as good as antibodies, but you don't have antibodies for several days afterwards. This is coming up, at sometime in day 2 of an infection. Okay, so they do 2 things. They cause opsonization, and then they also will fix complement. Okay, here's C-reactive protein, and here's mannose binding lectin. This looks like a little antibody, but they will fix complement, and I'll show you that in a second. C-reactive protein is a marker for. Wait, one second. Is a marker for inflammation. This is the lectin pathway. We have mannose binding lectin being produced, so you get activation of complement, and here you get C-reactive protein. So these are the other 2 pathways that I spoke to you about. But an important thing is that C-reactive protein is a marker of inflammation. Now you know how it works. And Dr. Bakshi mentioned that, and he showed you that it was a prognostic factor in COVID disease before we really knew too much about it. When C-reactive protein was going up, we knew the patient wasn't doing well. C-reactive proteins going down, we knew the patient was getting better. Here, patients coming in with a 10.74, and it's released, and it's 1.05. So C-reactive protein, which is made by the liver due to infection, okay, now you know why we measure it, okay? Now, we're just going to finish up now on, viruses. Okay, so we've been talking about bacteria. Bacteria do a good job with the, being taken, care of with NF-kappa B and that sort of inflammation. Here's viruses. Viruses, you need interferons. Here's a virus coming in. I put in the toll-like receptors and the stats. And what happens is it'll come in, you get a transcription, okay? And the interferon beta is what's going to be made. Interferon beta gets secreted, okay? So a virus comes in, we have the toll-like receptors or the cytoplasmic molecules that will bind to the virus, whether it's the double-stranded RNA, single-stranded RNA in certain conformations, and we begin to produce interferon beta. What happens is this interferon beta could bind back to this cell. It's in an autocrine response, or it's in a paracrine to another cell. And what happens is now you begin to make interferon alpha. So the virus induces interferon beta, and interferon beta will then induce interferon alpha. Okay, for the most part, this virus-infected cell is a goner, okay, because it's already been infected. But what you want to do now is you want to keep the normal cells alive. And that's what you're going to do here with the, it's a paracrine effect to keep uninfected cells healthy. Next, what do interferons do? They increase resistance to viral replication. They prevent viral replication. They suppress the translation of viral RNAs. We'll discuss this soon. They actually increase MHC expression. But you'll understand that more tomorrow. I'm going to mention it tomorrow at this time, that this promotes a good cytotoxic response against viruses. They activate dendritic cells and macrophages. They activate NK cells. Those are natural killers to kill virus-infected cells, and they recruit lymphocytes, okay? Don't sweat over all of these effects. The MHC is important. Just know that it prevents viral replication. It increases MHC expression, and it does other things that will help generate the immune response. Okay, here I just wanted to show you that here the endosome toll-like receptors against different viruses, and sure enough, double-stranded RNA, single-stranded RNA, here you're going to be getting gamma interferon, interferon alpha beta and interferon beta being made. Okay, so that's toll-like receptors 7 and 3. Okay, and this is just showing you the intracellular molecules. This is a RIG1, and some viruses make uncapped RNA. Okay, and this is recognized as being foreign, so it's bound, and sure enough, you're going to get interferon production. Each one of these events that we've been discussing is a target for viral subversion. Here's coronavirus. Okay, coronavirus interferes with multiple steps during the initial innate immune response, RNA sensing. Here you have coronavirus molecules will prevent toll-like receptors, or the RIG1. Those are the cytoplasmic receptors. It also prevents the signaling pathway, okay? And it's also able to prevent the STAT activation also. So everything that we do to give us a good inflammatory response or a good interferon response, viruses know and they try to inhibit. So you have, and we'll go through this in a minute, you get a delayed innate immune response, longer incubation times, more viral spread. When the immune response finally kicks in, it's dysregulated, and that gives you the morbidity and mortality. We're going to finish on the NK cells, and then just one more point on the interferons and the NK cells. Natural killer cells are like T cells, but without a T cell receptor. There's no prior immunization. They're around all the time. They just need to be a little bit somewhat activated. They have multiple receptors. They're activating and inhibitory, and the balance of the ligands determines the outcome. When you read anything about natural killer cells, don't sweat the receptors. Just know some are inhibitory, some are stimulatory. Whichever you have more of is going to drive it in that right direction, okay? It's activated by interferons and IL-12, and it produces gamma interferon, which stimulates T cells. So listen to this. Interferons are being produced, right? So the interferons will stop the NK, they will stop the virus from replicating, right? Another thing that the interferons do is they will activate the natural killer cells, okay? And then those natural killer cells will then kill the virus-infected cells. Here are all these receptors. Don't spend any time worrying about them. Just know that when you have these stimulatory receptors, okay, it's going to activate the NK cell to kill the virus-infected cell. Some of them are inhibitory. For tomorrow, just remember that the histocompatibility molecules, or HLA molecules, are inhibitory. And again, so it's some are inhibitory, some are stimulatory. It's the balance of the two which really determines the outcome. One interesting point, we saw that when you have, at some point, you're going to have dendritic cells making their way into the lymph node to stimulate the immune system. Question is, when is the right time for that? Okay, does it happen all the time? Or, for example, is it only happen when a certain point is reached and we say, hey, the innate immune system really can't handle this infection, let's get the adaptive immune response. This is a very, very nice figure that was in your book, okay? Look what's happening here. You have NK cells and you have dendritic cells, right? The dendritic cells, for example, are infected with the virus. If you're going to be making a lot of natural killer cells, okay, a lot of natural killer cells, you probably don't need much of an adaptive immune response. It should be able to handle it. And you have dendritic cells, by the way, they're infected with viruses. Most viruses can infect dendritic cells, okay? Some of them can't, okay? But this is virus infected, so natural killer cells, okay, will be stimulated, but then they will actually kill the dendritic cell. So if you're going to get a ton of dendritic cells, they will actually kill some of the cells that are stimulating them, okay? And those dendritic cells won't need to make their way to the lymph node and stimulate the T and B cells. But if you have a situation where you have very, very few NK cells, there are not enough NK cells around to kill the dendritic cells will make their way into the lymph node and stimulate the adaptive immune response. Okay, just to finish off now, this is, here's virus titer. Okay, virus starts replicating, interferons start coming up. Okay, that's from like day 1.5, you start getting good amounts of NK cells coming up. The virus titer starts to level off, but it doesn't go anywhere. Then you need antibodies and T cells to come in to get rid of the virus, and then you're going to get rid of the virus. So this is the innate immune response against viruses. Here's the adaptive immune response. What happens at COVID? This is the interferon response. Instead of it being in the beginning, it's delayed, okay? And that you're getting a lot of replication of the virus, and then that gives you the severe immunopathology that you have, okay? Because by the time the immune response kicks in, you got tons and tons of virus, and it really messes things up. So this is the last slide. An immediate immune response. If the pathogen is recognized by preformed effectors and tissue-resident effector cells, okay, you can actually eliminate the infection, okay? If it's still it's not totally under control, you can get the induced innate immune response, okay? Let's say the macrophages and the dendritic cells were not enough. Now you're going to get neutrophils coming in and C-reactive protein, and maybe that will help eliminate the immune response, okay? This is 4 hours to 4 days. Then what's going to happen is if this doesn't work out, then you're getting the adaptive immune response. You get T and B cells, and hopefully that will result in elimination of the infection. Okay, thank you very much. It's been a long day. And see you all tomorrow.